My name's Quayle, and I'm currently a postdoctoral research associate working in the Department of Oncology and Metabolism here at the University of Sheffield. My research interests focus on intratumoral heterogeneity and how specific subpopulations of tumour cells are responsible for chemoresistance and tumour recurrence. My PhD project was focused on developing a novel preclinical model for the identification, isolation and characterization of mitotically quiescent quiescent tumour cells thought to be responsible for both de novo drug resistance and the initiation of metastatic disease in breast cancer. So using a fluorescent lipophilic dye retention method, I was able to identify a latent quiescent subpopulation accounting for less than 2% of the total population in multiple human breast cancer cell lines of different molecular subtypes. The relatively quiescent nature of these dye-retaining cells was confirmed by their tendency to collect in the G2M phase of the cell cycle and by a significant enrichment with cells that were Ki67 negative or in G0 phase of the cell cycle. On immunoprofiling of the quiescent cell population, we illustrated a partial overlap with the CD44 positive, CD24 low signature that's commonly used to identify breast cancer stem cells, but we also showed a significant enrichment for cells that were CD44 positive and CD24 positive, as well as being positive for aldehyde dehydrogenase. These quiescent cells also displayed an enhanced ability to survive chemotherapy and exclusively contains subclones capable of subsequent outgrowth. So the current project that I'm undertaking aims to define the critical biological mechanisms that underpin the generation and survival of this cell population and which function to mediate cell survival during systemic dissemination and following chemotherapy. The ultimate aim of my work is to identify novel therapeutic targets that will lead to the development of improved therapies for metastatic breast cancer.